Hi, I'm Jules, and today we are going to talk about viral life cycles. The image you see here is a virion, also called a virus particle, of SARS-CoV-2, the virus that causes COVID-19. On the outside of the virion, you see the S or spike protein, which plays a key role in allowing the virus to infect cells. This is why vaccines against COVID-19 are designed to train the human immune system to recognize the S protein. The key topics we will talk about today are how to classify different types of viruses, what are the key steps in most viral life cycles, how these steps are carried out in the SARS-CoV-2 life cycle, how those steps may be carried out differently in other viral life cycles, and then a brief summary of key takeaways about viral life cycles. So let's talk about how we think about different types of viruses. Here's some key aspects that can help us characterize how we think about a virus. First of all, what is the specific shape of an individual virion or virus particle? The three main categories that we can use are usually helical, also called broad-shaped, like the rabies virus, or spherical, like the herpes simplex viruses, and a broad category called complex, filamentous flaviviruses such as Ebola, or bacteriophages, which are known for their head and tail spider-like appearance, but can include other diverse shapes. We can also ask, does the virus have an envelope? A viral envelope is an outer lipid membrane derived from host materials like the endoplasmic reticulum, the Golgi apparatus, or the plasma membrane. Some viruses have evolved to have an outer membrane because it makes it easier to infect host cells, which have membranes, but others are non-enveloped. This is also called a naked virus. You can see in the diagram in the upper right that all viruses have genetic material in the center of their virion, and it is protected by a protein shell called a capsid. For naked viruses, capsid proteins are their primary way of interacting with cells and the environment. But for enveloped viruses, that role goes to the envelope. Typically, the envelope will contain glycoproteins to aid in infecting cells. Another way we can classify viruses is by the content of their genome and how that genome is processed in the host cell. This diagram shows the Baltimore classification system, which is a useful tool for comparing how and why viruses replicate in specific ways in their host cell. In this system, viruses are divided into 7 groups, each based on the type of genome and how that genome is processed into mRNA that can be translated by host machinery. Firstly, is the genome DNA or RNA? Is it double-stranded or single-stranded? Is it plus sense or minus sense in terms of complementarity? What intermediate steps are needed to make lots of mRNA which can be translated into viral proteins? There isn't enough time to dive into each class, but let's look at the group that contains SARS-CoV-2, which is group 4. Viruses in group 4 have a single-stranded plus-sense RNA genome. Some of the plus-sense RNA genome acts like mRNA and can be immediately translated. However, the virus still needs to make more copies of its genome, so you can see that there is an intermediate step to make minus-sense RNA, which could then be replicated into more plus-sense mRNA to make viral proteins. There's more details to the steps, which I'll go into later. Group 6 contains retroviruses like HIV. These viruses also have several intermediate steps to replicate their genome, most notably a reverse transcription step in which RNA is used to produce DNA. When we look at SARS-CoV-2, we can classify it by its shape and structure. It is a spherical virus with an envelope. Its genome, which contains plus-sense single-strand RNA, as discussed in the previous slide. In this diagram, we also see some of the virus's key proteins. We see the S-glycoprotein sticking out of the envelope for all receptor binding with host cells. We also see the enzyme hemagglutinin esterase, which acts as a glycoprotein that aids in attachment by binding to sialic acid residues on the host cell membrane. We see that proteins E and M are embedded deep in the envelope. These proteins are mostly involved in virion assembly in the host cell. In the center of the virion, the N protein makes up the capsid protecting the RNA genome. When we look at viruses in general, there are some unifying characteristics to their life cycles. In order to use the cell to replicate itself, the virus must do the following: attach to the host cell, penetrate that cell in order to introduce its genetic material and any necessary proteins, biosynthesis to make new copies of its genome and any necessary proteins needed in order to make new virions. Then the new virions must be assembled, and finally the new virions must be released from the cell in order to infect new cells. So how do these steps occur in the life cycle of SARS-CoV-2? In the enveloped virus life cycle, the virion attaches to the cell, usually through receptor binding. Penetration usually occurs through receptor-mediated endocytosis or fusion with the cell membrane, and the viral contents then can enter the cell. Inside the cell, biosynthesis occurs through replication of nucleic acids and translation of viral proteins. And after assembly of new virions, the virions must get coated with membrane from the host organelles. And finally, new virions bud off from the cell membrane and are released from the cell to infect new cells. When we look at the steps of attachment and penetration in SARS-CoV-2, we see that the S protein binds to the ACE2 receptor on the cell's membrane. This allows entry via receptor-mediated endocytosis. During receptor-mediated endocytosis, the protease camptothecin L aids in fusion of the endosome and viral membrane to allow escape of viral contents. The bond between S and ACE2 is strong, which means it requires fewer virus particles to attach to the cell and attempt penetration. Entry can also occur via fusion. Another protease TMPRSS2, aids this process by cleaving the S protein to allow for faster fusion and emptying of viral contents into the host cell. After attachment and penetration, starting at step 2 in this diagram, we see that the virus has penetrated the cell and released its viral contents. In step 3, the RNA genome is translated by the ribosome in order to make proteins needed for viral replication. The arrow going from step 3 straight to step 5 shows that some of that RNA will later act as a template for transcription of the subgenome. Going back to step 3, the proteins that are translated are actually large polyproteins, and they have to be broken down via proteolysis, we see here in step 4. Upon translation, 3CL-Pro, a protease, becomes active and autocleaves itself from the rest of the polyprotein. 3CL-Pro then cleaves the polyprotein and allows other proteases to become active to finish cleaving the polyprotein into its functioning parts. These functioning parts make up the replication transcription complex and include RNA helicase and RNA-dependent RNA polymerase and proteins for dividing a 5' cap and poly-A tail on the viral RNA to prevent host recognition. In step 5, the replication transcription complex makes more RNA for virions, but also performs necessary transcription. So it is both replicating and transcribing. This is because SARS-CoV-2 has both a viral genome containing genes for structural proteins and a nested subgenome for the non-structural proteins N, M, E, and S. While the viral genome is immediately translated and becomes the replication transcription complex, the non-structural genome, or subgenome, must be transcribed from the original viral genome template. This is accomplished by skipping or frame shifting on the viral RNA template. After the subgenome has been transcribed, these transcripts are translated and processed in the endoplasmic reticulum in step 7. Then assembly, also called packaging of the proteins in viral genome into new virions, occurs in the Golgi apparatus in step 8. The M protein organizes the assembly of new virions, while the N protein specifically packages viral RNA into a ribonucleoprotein structure within the capsid. The new virions are coated with a new lipid envelope derived from the host cell. In step 9, the virions are then released via exocytosis. Protein E is known to facilitate budding by causing curvature in the cell membrane during this step. And then the virions go on to infect new cells, and the life cycle begins again. There are some other specific life cycles for viruses you may have heard about. For example, we talk about naked viruses that don't have an envelope, so they can't fuse with the cell membrane when entering or exiting the cell. This can make them tougher to kill because they have evolved to have extra tough capsid proteins to compensate for not having an envelope. They also have no antigens on their surface for cell-mediated immune responses. Some naked viruses also can only exit the cell via cell lysis or violent cell death, and this aids to their virulence. Examples of important naked viruses include norovirus, poliovirus, rhinovirus, hepatitis A, and HPV. Now we look at bacteriophages. These are viruses that target bacteria only. They are also naked and usually kill cells via lysis. When this happens, it is called the lytic life cycle. However, some are known for the lysogenic cycle, where the virus integrates into the host genome. Host cell division will spread the viral genome until some of the viral DNA leaves the genome and begins again the lytic life cycle route. You may have heard of the bacteriophage T7. It is a lytic phage that infects E. coli. The T7 promoter of this bacteriophage is used in molecular biology because of its high affinity for the T7 RNA polymerase, resulting in high levels of expression. Retroviruses, like HIV, target animal hosts only and are known for their unique genome processing, in which their RNA genome is reverse transcribed into DNA using the viral enzyme reverse transcriptase. The complementary cDNA made through reverse transcription will then be integrated into the host genome, making what is called a provirus. The provirus will be expressed along with the cell's genome. However, sometimes the viral genes will not be transcribed and translated along with the host genome due to low levels of needed transcription factors or other cytokines or proteins that trigger this. This dormant state is referred to as latency. However, similar to the lysogenic life cycle in bacteriophages, if the infected cell divides, it still spreads the provirus genes, and they will someday be able to go actively replicate the virus. Latency therefore hides the virus from the immune system. Antiretroviral therapy for HIV keeps the virus in the latent stage. Here are some key takeaways about viral life cycles. Viruses can be classified by their shape, whether or not they have an envelope, and their genome, and how it must be processed in the host cell to become translatable plus sense mRNA. For example, SARS-CoV-2 is a spherical, enveloped, plus sense single-stranded RNA virus. All viruses have similar life cycle steps, which include attachment and penetration of the host cell, biosynthesis to replicate the viral genome and synthesize viral proteins, assembly of the replicated genome and proteins into new virions ready to infect cells, and release of these virions from the infected cell. During the life cycle of SARS-CoV-2, the S protein aids in attachment to the cell, and penetration, also called entry, occurs through fusion or receptor-mediated endocytosis. Biosynthesis occurs at both the genome and subgenome level. Immediate translation of the viral genome by ribosomes produces polyproteins that have to be cleaved to make the replication transcription complex, and then the subgenome has to be transcribed and translated in endoplasmic reticulum to produce proteins S, N, E, and M. In the host cell Golgi apparatus, proteins E and M aid in assembly, and protein N forms a protective capsid around the viral genome of new virions. New virions are then released via exocytosis to infect new cells. SARS-CoV-2 follows an enveloped life cycle style. However, other kinds of viral life cycles include naked viruses, which have no envelope and therefore can be more virulent because of their tough capsid and/or lysis of the host cell, bacteriophages, which can exhibit lytic cycles lysing the host cell, or lysogenic cycles in which they hide and replicate in the host genome, retroviruses, which use reverse transcription to go from RNA to DNA and incorporate into the host genome, leading to periods of viral latency where viral proteins are not translated but hide in the host genome. And that concludes this lecture on viral life cycles.